Thank you all for allowing me to speak in this forum. I'm really excited to, get a different perspective on the stuff that I'm doing. What we focus on in the Tsukiyama lab is understanding how cells go from or my focus in the Tsukiyama lab is to understand how cells go from this really repressed quiescent state to a really active state during cell division. So I'm going to be talking to you more about like kind of one-dimensional nucleosome. We're going to be looking a lot at nucleosome positions today, so it's a little bit of a different perspective. But what is quiescence? So a lot of us think about the cell cycle where we have G1 and then S phase, where we have DNA replication, and then G2M phase, but there are actually other stages where cells can exit the cell cycle, where basically these are considered G0. We have two different major types of G0, and irreversible G0, which can include like aged senescent cells or terminally differentiated cells. And then we have an irreversible or a reversible G0 where the cells are considered quiescent. So these can actually go into quiescence for like this is a universal phenomenon. Bacteria can go into quiescence. Our stem cells can go into quiescence. And in the case of microbes like bacteria and yeast, eukaryotic microbes, this is really important for like long-term survival. You can actually like keep these cells in water for a year and then activate them, even more than that too. But in terms of humans, it's very important for maintenance of the stem cell niche. We have quiescent stem cells in the brain that we have to be able to activate during like brain injury or stress. So it's actually a really important phenomenon. We focus on yeast, which can enter quiescence by the main way we do this is by culturing the cells for 7 days in rich medium, and then we get a heterogeneous population. And what the advantage of being able to use yeast is that we can purify the yeast over a Perkel gradient, and the yeast that are quiescent go to the bottom of the gradient because they store a lot of different sugars and such, and we can get rid of the non-quiescent cells. So we can really like purify this population. And an emerging hallmark of quiescent cells in both yeast and mammals is that there's a more repressive chromatin environment. And I think this is going to be really of interest, and I'm hoping to kind of give an overview of this and kind of hopefully convince people that quiescence is like a cool system to kind of study the high dynamic range of chromatin environmental changes and how they affect DNA templated processes. So one of the biggest findings on quiescence and yeast specifically was done by a former postdoc in the lab, Jeff McKnight, who was an amazing scientist, and he pretty much spearheaded all of the work that we do. He found that basically the entire genome is completely removed of acetylated histones, so they could become hypoacetylated. Then promoter regions upstream of genes, the nucleosome depleted regions are more narrow. There's higher histone occupancy, and there's little to no transcription in this process. And then others in the lab show that this chromatin also becomes more condensed as well. My question is, how do cells go from this really repressed state to this active state? How do they turn on their transcription is going to be the first part of my talk. And then at the end, I will talk a little bit about DNA replication for a work in progress project. So we were really surprised by this finding that transcription reactivation can actually occur within minutes of giving this yeast food again. So you can see here in the quiescent state, if I'm looking at this is Pol2 ChIP-seq data, I just did a k-means cluster to kind of illustrate the changes over time. What I hope you can appreciate is that there's a huge increase in this Pol2 occupancy within 5 minutes of refeeding. And we see this similarly with nascent RNA-seq, as well as just doing a Western blot on phosphorylated Pol2 where it's activated. So you can really see this difference happening really quickly. And so my main question is, how do cells do this? How do they switch so quickly when their chromatin is really repressed? So one of the obvious things I was thinking of, like, oh, well, maybe there's like acetylation that occurs really quickly, but it actually doesn't, it increases a bit, but it actually doesn't really increase nearly as much as we see for RNA or for Pol2 transcription. So I don't think the acetylation is actually going to be driving this process. So what do we do? How do we look at this? So what we were really interested in was the nucleosome movement and how chromatin accessibility changed. So basically, I took advantage of using a low digestive MNase and a high digestive MNase to look at the relative resistances of the nucleosomes. So at a lower digestion, we can pick up some more weaker particles in the nucleosome depleted regions. And what I hope you can appreciate is that between quiescence, which I will be calling Q throughout this, and 10 minutes, we see a pretty dramatic increase. This is averaged over 6,000 genes. So this is a pretty dramatic decrease. And you can even see shifts over the gene body, which is likely due to transcription disruption of the nucleosomes. And what are the factors that are going to be opening up promoters? Well, these are going to be most likely ATP-dependent chromatin remodeling enzymes, which are a class of enzymes that are used to basically organize the chromatin. And so these can be involved in nucleosome assembly, chromatin access, which SNF is the major player that I will be talking about, and then also like exchange for different variants, for example. RISC is a type of switch SNF chromatin remodeler. It's known as the NDR creator. It can slide and eject nucleosomes to create nucleosome depleted regions. In humans, and it's known as PBAF. In flies, it's known as polybromo. And it's mutated in 20% of all cancers, and it's highly studied to try to understand how chromatin access is regulated. So I wanted to know, okay, well, maybe RISC is regulating quiescence exit because of that change that we saw in the MNase digestion profiles. So I basically wanted to look at how RISC was bound in quiescence. And what was really surprising was, so if you look at its classical binding profile using ChIP-seq, RISC is kind of like around like the -1 +1 +2 nucleosomes. But in quiescence, you can see RISC is like really like bound to nearly all promoters in quiescence at the NDR. And for those of you who are RISC and switch SNF experts, this is in contrast to the way people sometimes are using techniques to look at RISC binding, which is like check-seq or cut and run, where they do see RISC binding in the NDRs at wider NDRs, but this is quite extreme. We see it even at the more narrow NDRs too. So this was really interesting, and I'll get back to this at the end. But so basically I wanted to see, okay, if we deplete RISC in quiescence, what happens? We see NDRs to narrow, nucleosome positioning in gene bodies will weaken, and then histone occupancy will increase in the NDRs. And these are all classic phenotypes of RISC depletion in cycling cells. So does RISC promote transcription during quiescence exit? I don't have time to go through all of the data, but I do have this published in eLife from last year. And what we saw was that RISC changes or is responsible for regulating the transcription program in 4 ways. And basically, when we get rid of RISC, we see a lack of PAL2 initiation, PAL2 elongation defects, PAL2 upstream start site selection potentially, or cryptic usage of cryptic promoters, as well as antisense transcription goes up. But one thing I do want to highlight is that we see RISC in NDRs in quiescence, and then we can actually watch RISC movement over time. And this is in that paper. So we can see RISC getting activated and move the nucleosomes. So I think this is a really cool way to be able to see how ATP-dependent remodeling enzymes are activated genome-wide. So I think this will be a useful system for people as well to look at. So what about other regions of the genome? So I'm going to switch gears here and talk a little bit about work in progress project on DNA replication. So I wanted to kind of highlight, we've just talked about transcription start sites, but the nucleosomes around origin sites, so these ACS sites, which is what we call them in yeast, you can actually see the MNase resistance is quite high in quiescence compared to log cells. And this is again data from Jeff McKnight's paper. So what happens during quiescence exit during at these origins? And so actually nucleosome positioning around origins does happen. It doesn't get back to log phase during, which is the cycling phase, during this first G1 event. But you can see there is more positioning, and the NDRs are a bit wider. And you can see this happening here. And then you can even see like the +1, +2 nucleosome shifting even more as cells are getting ready for DNA replication. So what is responsible for moving these nucleosomes? Well, we screened the entire, all of the ATP-dependent chromatin remodeling enzymes, and we did not find anyone that actually is responsible for binding to these origins in quiescence or in the first G1. So this actually led us to start thinking about the pre-replication complex itself and how this could be involved in moving these nucleosomes to prepare for DNA replication. And highlighting this idea, the origin recognition complex, ORC, there's a recent really nice paper that actually showed that ORC itself has an intrinsic ATP-dependent nucleosome remodeling activity in vitro. And ORC is known to bind the nucleosome. It's known as a nucleosome disrupting factor, but this is one of the first times where we can actually see disruption of H2A and H2B. So here's just a nucleosome eviction or a histone eviction assay where they basically took ATP the nucleosomes and added ORC. And you can see it can evict H2A and H2B in an ATP dependent manner when they use an analog and no longer can evict. So this is really interesting idea. So I'm looking into how ORC could be doing this in vivo. And so one interesting thing we saw was that ORC in quiescence adapts this like lower binding, maybe even a more restricted binding in quiescence, and it kind of spreads out a little bit. We're going to do higher resolution mapping of ORC to further define this. But if we sort nucleosomes based on the increase of ORC bindings, we have 3 clusters now. We can see that the origins where there's the most increase of ORC binding, we can see the nucleosomes repositioning more compared to ones where ORC was already bound in quiescence. We can't ignore MCM. This is also known as a nucleosome disrupting factor, though not ATP dependent. And so we do see an increase in movement of nucleosomes upon MCM bindings, particularly at a middle cluster here. And in quiescence, there's actually no MCM binding. So this is also another important candidate for nucleosome repositioning in quiescence exit as well. And so we're currently testing these models. So this is very much new data. So we are a new project, so we can actually test whether ORC alone or ORC plus MCM can reposition these nucleosomes. And we're taking advantage of oxen degron systems and mutants to answer these questions. And so to summarize this, I told you today quiescent chromatin is more restrictive to both transcription and DNA replication as cells re-enter the cell cycle. RISC is the key regulator for this huge burst of transcription that occurs during these early quiescence exit events. ATP-dependent chromatin, classical ATP-dependent chromatin remodelers are unlikely to be involved at replication origins during exit. And we're testing the model that pre-RC members can reposition nucleosomes during exit. But what we're learning is that quiescence to growth transition or quiescence entry itself can be a powerful system to study these dramatic nucleosome repositionings across DNA templated processes. You could probably also look at chromatin folding. There's Sarah Swigert, just started her lab at Colorado State, and she's looking at chromatin folding during these during quiescence entry. And so I think there's a lot of really cool questions that we can address in this system. With that, I'd like to acknowledge everyone in the Tsukiyama lab. I'm also doing some work with Patrick Pattison on quiescent neural stem cells at Fred Hutch. And yeah, if you have any questions, I'm happy to answer them.
Thanks, Fen, for the talk. So I have two maybe naive questions. I don't know, but I mean, one is regarding this very fast activation of the genes.
Mm-hmm.
So how the POL2 is stored during quiescence? I mean, it's stored in the cytoplasm or it's in the nucleus?
I really want to answer that question. I do not have that data because I haven't looked at imaging or anything, but I would love to be able to look at that if anyone is interested in collaborating or something. Because yeah, one of the ideas I had was like maybe or not or Pol2 could be like kind of sampling the genome very like unstably binding and like maybe able to activate really because it's so rapid. Or maybe there's phase separation involved. I don't know, but I would love to like answer that question because it's so rapid. Um,
And the second question that I have is, I mean, again, regarding this very fast activation. So do you have, because all the genes are expressed rapidly, or it's just a few genes that are
it's about 60% of the genome is significantly increased transcription, and then there's like lower levels of increase. So it's a lot of the genes. It's like this huge event, basically.
Okay, because do we have evidence about how RSC is targeted to the +1, or I mean to the TSS? I mean, what is the mechanism that target RSC? And again, is there any priming mechanism that is doing that?
Yeah, I'm very, very interested in that question. In fact, I'm applying for jobs right now, and I'm proposing to kind of study that. So we don't know yet how it binds. I've done some biochemical assays looking at post-translational modifications on risk in quiescence compared to log. They are very different. And so I think that there's going to be some kind of post-translational modification involved in this system. There are also very like different subcomplexes of risk that I think might be forming. I have some preliminary evidence of that too. So yeah, that's going to be a huge question that I'm really interested in answering, but I do not have the answers yet.
Sorry if this is, maybe I just disconnected for a second, but so you were showing us that when you deplete risk, you get this effect on positioning of nucleosomes, but did you show us what happens to transcription itself?
I didn't show it, but I do have it here. So for this, we can see genome-wide, when we get rid of risk, we see a reduction in Pol2 occupancy over time. And so what I showed earlier is we see this like really strong increase in the beginning of quiescence exit, but then it actually goes down a bit. So I think there's like this hypertranscription event that occurs, and then it has to be toned down in order to get ready for DNA replication. But this peak in transcription never actually occurs when we get rid of risk. So the transcription is actually pretty abrogated. And I can just like quickly show actually when we get rid of risk, you can even see like if we take the ratio of the transcripts, you can see a lot more transcription happening even upstream of genes. And then also we get a lot of antisense transcription. So the whole transcriptome kind of gets messed up because there's a combination of Pol2 having really strong activity, and then the nucleosome movement also, actually you can even see an example here. If we look at risk binding and this one gene example, PTP3, you can see in wild type, you have this like nice start site selection here, and then you have upstream transcript, and you can actually directly look at the nucleosomes and see that this in wild type cells, you can see there's this huge nucleosome depleted region that's gone without risk, and then upstream of that, there's a new NDR. I don't know how this new NDR forms. It's another question I have. But then Pol2 will basically go where it can. So the genome is very destabilized, or like the transcriptome is very deregulated during this process.
But when a cell is quiescent and you deplete risk, nothing happens, or?
Yeah, so we can deplete risk. We do see a reduction in Pol2 binding. The little Pol2 that binds, there's still a bit of a reduction. I don't know how quantitative this, I mean, I did spike control, but I don't know, it's such a low level. But we still get quiescent cells. So the way I deplete risk, I deplete it after the committed step during quiescence entry. So that kind of helps too. But yes, so.
Just wrote, okay. Does some nucleus swell in these 5-10 minutes?
I would love to answer that question if anyone wants to collaborate. I would love to look at that because this you would imagine it probably would, but I have not looked at that. That's actually, I'm kind of excited to be in this forum because we can maybe, get new ideas to look at these things.
You need to have it because it is a critical phase transition that is occurring.
Right.
Cannot be otherwise. Yeah, that's one. And the other question, by the way, I've read about G1 initial on bivalency activation of stress response genes. Is not anything of this kind involved here?
Bivalent, like bivalent histone modifications?
Yeah, histone, modifications, K4 and K27. Usually 4 is activated, and it is characteristic just for the beginning of G1 phase.
Right. Budding yeast do not have polycomb or histone K27 acetylation. So actually, it's actually kind of interesting. So I think what keeps them poised is risk binding, and that inactive risk ready to go. But we haven't really looked too much at histone modifications. Marianne Osley's lab has done a lot about that, but a lot of them acetylation, it's gone in quiescence, and I think that's directly linked to the metabolic state too, because these are starved cells as well. So I think cells have to be able to activate genes without acetylation marks too. So
the next question is from Leonard Collin. Very nice, Christine. Do you know if TSA treatment can affect entry or exit of quiescence?
Yes, so I know you can treat cells with TSA. I can't remember what the quiescent cell yield ends up being. You can, if you delete RPD3, which is an HDAC for these marks, you get very small amounts of quiescent cells because they need to get rid of the histone acetylation in order to enter quiescence. For quiescence exit, I did treat them with TSA and it didn't really have much of an effect on exit that I could tell, but I haven't really dived too carefully into that. That's a good question.
I have maybe a more general question. Would it be possible to make like a genetic screen to perform an unbiased screening of factors that are required for regulating quiescence? How would you do that?
Yeah, I would love to do that. So there's a lab here at the Linda Breeden's lab. She's done a lot of really great work with the yeast deletion collection and looking at that. And you can she's done like a ton of basically flow cytometry assays to look at quiescence exit. And we have some collaborations with her too, and we're actually following up on some interesting leads there. But yeah, it's something that we've definitely thought about. And with the yeast strains, we have like the whole deletion collection, so we can very not easily, but like more easily than maybe some other systems look at these things. So we have been doing a little bit of that in the lab as well, but I would love to come up with more screening ideas. For mammalian system, we have a reporter that can that increases in quiescent cells, so we can use a flow sorting mechanism there and use like CRISPR screens too. So we're actually actively thinking about those as well.
So very, very nice talk. As usual, you're amazing. I have a very naive question. So when I see the waves, when you are activating the exit from quiescence, there is this wave, there is the blue signal, I guess. I guess that is where there is the pole 2, right? And eventually,
yes, like
and then it goes away. So when you remove risk, basically it's kind of more diluted, but it stays longer.
Yes. Yeah, and you can even see if you look a little closely here, pole 2 has like 2 peaks. So I think that's pole 2 getting stuck or going in the wrong direction. And so we see a lot of antisense transcription happening too, as well. And then also pole 2 itself, when we get rid of risk, I think it's this pink line. Yeah, this pink line here, it gets stuck too. So if we zoom in on a subset of genes, POL2 kind of normally in the dark blue, we see POL2 like having its like peak at the +1 and then it can go through. But here POL2 gets kind of stuck. And so I think when we zoom in at that 30-minute time point here, that's what we're seeing.
But then what does it mean? Is this like, a sort of a slowdown in the processivity of the POL2? Or it
I
just...
think so. Yeah, I think so. I would love to look at like do ProSeq or something.
Yeah,
to
exactly.
Look at
That
yeah,
would be very cool.
Yeah, I have so many questions. I need more hands to answer all these questions
If
because I do think that
Yeah.
risk will travel with POL2 and basically help it get past this +1 nucleosome. And I actually do think that risk and POL2 directly interact. There's a really nice paper from Julie Suterina many years ago where she showed that one risk subunit can directly interact with all 3 polymerases. So it's definitely something I'm thinking about for the future.
So still in the plot that you showed before, not the slide before with the blue, the genes are obviously like all aligned, right? The line that is on the plus risk, it corresponds to the line on the other side, or they are only put it there by the most prominent in terms of...
It's been a few years since I made this, but I'm pretty sure they're all linked
Okay.
across.
So basically it's kind of a proportional kind of effect. The more the POL2 is there, the more it's also staying upon the removal of risk. It's not that there are any kind of genes that are jumping, for example, from one state to the other.
Right. Yeah, that is a good question. So there are genes that are more resistant or resilient to risk depletion, and those are ones that have really wide NDRs to begin with, and have they're like these pioneer type factors like REB1, RAP1. Those types of genes are the ones that will be more able to withstand risk depletion. But then there are some, I haven't done I don't think I have it in here, but there are some transcripts that increase when risk is gone. But the most part, it's usually non-coding or antisense transcripts, because as an example, I don't have specifically here the antisense transcript, but what happens, the phenomenon that happens is that there are some NDRs that get created in the absence of risk. And those, basically POL2 will go to those.
Yeah,
And
to be honest, when you show this, that's exactly what I was thinking, that maybe that this kind of decrease just because there is antisense, but that specific example, it shows that just transcription is starting a little bit earlier in terms of base pairs, but still going to the same direction because there is no so much transcription on the opposite side. So that's why I was so. A stupid question, Christine. So in mammalian, basically from cage experiments, what they've shown in the Phantom project is that different promoters, they have different way of starting the RNA production. Some promoters are much more precise, which it means that literally every promoter is firing exactly from the same base, but there is other promoters that show a little bit more variance, like, there can be a couple of bases before, a couple of bases after. I wonder if this is happening also in yeast. And if that's the case, it could be the different geometry or like whatever architecture of the promoter, call it whatever is better. It may actually show some different pattern in the way in which they depend by risk.
Absolutely. Yes. And that's actually definitely something I'm really interested in, like kind of diving more. This is kind of more of a survey of all the responses of risk depletion, but I would love to get more zoom in more on those different aspects, because I think it would be really interesting, and then see what transcription factors are required for that too. Yes.
Thank you very much. Awesome.
Yeah, thank you for all the questions. It's cool to think about.